Okay, thank you all for the opportunity to present today. I am also in the Department of Molecular Genetics and Microbiology. I have a joint appointment in obstetrics and gynecology, and I'm a member of the UNM Cancer Center as well. Our technology is focused on human papillomavirus infections, and human papillomaviruses are part of the normal microbiome in humans. We're essentially covered with HPVs. To date, we know that there are over 200 or 250 genotypes or strains of these viruses, but in most cases, they don't cause any evidence of infection. But a subset of HPVs can typically cause papillomas and warts at virtually any surface of the body, of the mucous membranes and the skin in the body. And many of these papillomas or warts can progress to cancers. HPVs are the most common sexually transmitted pathogens in the world. And importantly, they cause nearly 5% of the total cancer burden in the world. This includes almost 100% of cervical cancers, other anogenital cancers shown in pink, and a rising subset of head and neck cancers in humans. You may know that there are safe and effective vaccines against HPVs that protect from the most medically important HPVs, the sexually transmitted and cancer-causing HPV strains. But vaccine coverage in the United States is unfortunately fairly low. The vaccine target age is 13 to 17-year-olds before sexual debut, and we hover around 50%. But in developing countries, these statistics are even lower, because developing countries don't have the infrastructure or can't afford the vaccines, and distribution among candidate vaccinees is quite low. And because of this, and the fact that many people have already been infected with HPVs, the prediction from the World Health Organization is that HPV-related cancers will be on the increase until the year 2040. And so this presents a serious unmet need for effective and affordable ways to prevent HPV infections in populations that are at risk, that need to be vaccinated or that cannot be vaccinated. And that is really the topic of some of the research in my laboratory. So there are a number of places where you can target viral infections. Viruses have to complete a number of steps to establish infection, beginning with binding to host cells, entry of the host cells, and often this is by endocytosis. The virus has to be trafficked to the appropriate site so that it can release its nucleic acid to the appropriate cell compartment to begin genome replication. And the attachment phase is really the most attractive target, and it's the step that's prevented by most antibodies elicited by vaccines and by some antivirals. And so just as a point of where we're going with our technology, some viruses attach directly to specific cell protein receptors that can promote entry of the virus. Many other viruses and intracellular pathogens attach first to cell surface sugar chains, which then are thought to promote a handoff to a more specific receptor for entry. And for HPVs, these receptors on the cell surface are heparin sulfonated proteoglycans. So a protein core that has attached to it a number of heparin sulfate chains. The fact that these heparin sulfate molecules have a number of sulfate groups at specific sites imparts an overall negative charge to these molecules. And so this provides us with an opportunity to neutralize these viral binding sites. And we have chosen to use arginine-rich polypeptides, which are positively charged, to negate the charge of these heparin sulfate molecules and actually prevent the virus from binding to the cells. And we have tested a number of arginine-rich polypeptides, but today I'm really going to focus my talk on protamine sulfate, which is a naturally occurring arginine-rich polypeptide that is currently FDA approved for intravenous administration as a means to reverse systemic effects of heparin treatment. And so protamine sulfate, by virtue of its positive charge, can bind to the negative charges of heparin in the bloodstream to neutralize heparin's anticoagulant effects. And we wanted to test the ability of protamine sulfate in our model to actually neutralize these heparin sulfonated proteoglycans. And so we have several models of HPV infection that we use in the lab. One of the main models includes monolayer cell cultures of keratinocytes that are exposed to biologically safe HPV pseudovirions. These pseudovirions transfer a luciferase expression plasmid, and so we can measure infection in a safe manner by looking at luciferase outcome. So in this setup, we would treat our cells with the protamine sulfate prior to incubating with the virions, and then allow these to go a certain period of time. In this assay, we typically use 24 hours to detect infection. And what we can see in this graph is that when we add protamine sulfate to the cells prior to the virus being attached, at increasing concentrations, you get a dose-dependent decrease that's quite significant. Looking at the inhibitory concentration of 50%, we get a pretty substantial decrease at fairly low doses of this drug. We also tested this drug against our cells for cytotoxic events or cytotoxic effects, and show that there are no effects on the ability of the cells to replicate. So I forgot to mention in the previous slide that we had targeted HPV 16. That's the most common cancer-causing HPV, but we wanted to test a number of other HPVs that are involved in medically important conditions. HPV 5 causes a number of non-melanoma skin cancers. HPV 6 causes genital warts, and HPV 16, 18, and 31 are important causes of HPV-related cancers. And you can see that all of these viruses have a very good response to doses of protamine sulfate in this assay. We also used real infectious viruses in the laboratory, some that we can create in the laboratory and some that we can grow in organotypic skin cultures in the laboratory. And we show again that protamine sulfate pretreatment is effective at preventing these infections at fairly low doses. To take this into an in vivo model, HPVs are not able to infect laboratory animals, but we can use an established model wherein we can use these pseudoviruses as a proxy for infection in mice. And so this is a vaginal challenge model or a cervical vaginal challenge model that has been shown to be effective in testing HPV vaccines and a number of antivirals in this system. And if we look at mice that have not been pre-treated with protamine sulfate, we can see, if we compare them to those that have been treated with protamine sulfate before they're infected, that we get a substantial and statistically significant drop in infection, showing that this also works in an in vivo model. We looked mechanistically because our hypothesis was that this drug would prevent HPV attachment to cells. And by confocal microscopy, that's what we saw. If we have viruses that are labeled in red, when we pre-treat the cells with protamine sulfate, we get much reduced binding to the cells. We can also look at this in a biochemical assay, looking for the major capsid protein of the virus, where when we treat with protamine sulfate, we get a reduction. And if we look over here at biological replicates, we get a significant reduction in binding to the cells. And we wanted to use this assay because the mouse vaginal tract is present, has a fairly neutral pH, but the human vaginal tract tends to be much more acidic. And so we wanted to test to see if this treatment would hold up under an acidic environment. And so we used the same binding model. So in the presence of a low pH, the protamine sulfate is still able to prevent binding. And if we look over here at replicates, we can see that this is statistically significant. So this suggests that the protamine sulfate would still remain functional at a low vaginal pH in humans. We also wanted to look at potential means for delivery of the protamine sulfate. And a good candidate for this would be a moisturizer or a lubricant that is commonly used over the counter. So we tested hyaluronic acid. And what we found is when we complex or mix the protamine sulfate with low molecular weight or high molecular weight hyaluronic acid, that it still maintains its efficacy in preventing HPV infection. And so this suggests that it would be a useful delivery and easily functionalized with protamine sulfate for prevention of infection in humans. And so just in summary, we found that protamine sulfate, which is a drug that's already FDA approved for intravenous administration, is potent and a broadly active topical inhibitor of HPV infections. It's agnostic to HPV type, and we show that it works both in vitro and in preclinical mouse models. We found that this primarily functions by preventing HPV virions to interact with heparin sulfate molecules on cells. And this is particularly attractive for HPVs because it can confer activity against multiple HPV types. But the fact that it can bind and ionically interact with heparin sulfate chains on cells also makes it very attractive to prevent the attachment of other sexually transmitted infections that also require interaction with heparin sulfate. And there are a number of sexually transmitted infections that require this that are quite important human pathogens. And so I will close there. I do want to mention that this has been funded by an R01 to my lab from the National Cancer Institute to understand HPV infections. And the status of developing this is that we are interfacing with companies that manufacture hyaluronic acid, excuse me, or other moisturizers to try to partner for testing in humans. So I will finish there and take any questions. Thank you.